So thank you very much, Alexi, for this very nice introduction. And of course, I'm very honored to be talking after Professor Richard Anderson here and Nicolas Simon, who showed us already many very impressive BMIs in humans. So I will talk about the mouse model, and I will try to convince you that it's also interesting to do this kind of work in the mouse model, not only for advancing BMIs, potentially for humans, but also for understanding key brain mechanisms that underlie sensory motor learning. And everything I will show you has also been done by Luc Estebanez, and also with contributions from Dan Schulz, the head of the team, and Isabelle Firezo. So the introduction is partly redundant with Richard Anderson's introduction, so I will go quite fast, but just to recapitulate the ideas behind BMIs. So how do we learn a new motor skill? So quite obviously we need to practice many times. So if I want to learn to hit the right keys on the piano in the right order, I need to practice a lot. And as Richard Anderson showed, we also need to use sensory feedback to do such movements. And often we think about visual feedback first, but as he showed, we also need particularly somatosensory feedback for skilled movements. And so we saw already one example of with the woman who had her fingers anesthetized and had to light a match, and this was very difficult for her. There are other examples like Jan Waterman here, a patient who lost proprioception and touch as a young adult. And so this patient, so when he found himself suddenly without any tactile sensation, he was unable at first to move, really, even though he had full muscle control. But he was unable to stand up, unable to feed himself. And he had basically to relearn all of this by using visual feedback. And it was extremely difficult. It required always a very high level of attention for this patient. And so there was no voluntary fine effort movement that became effortless for him. And so these examples demonstrate really that somatosensory feedback is critical for doing fine voluntary movements with dexterity. It's also critical for learning new movements. And as I'll talk about briefly at the end, it's also important to allow embodiment, which allows users of neuroprosthetics to actually accept their prosthesis. And Zineb will talk about this later also. Okay, so that was for natural motor skills, but what about what about somatosensory feedback in brain-machine interfaces? So we saw already that some of the brain-machine interfaces that exist, like Professor Anderson showed, are based on visual displays, like this woman here in his lab learning to play the piano. But others are based on moving implementations like robotic arms. And we saw this patient who managed to drink a beer while modulating activity of neurons in the PPC and moving this prosthetic arm. So as Richard Anderson said, in order to have more efficient neuroprosthetics of this type, the motor part of the BMI, that is reading information from the brain and moving the prosthesis, is not enough, and we also need to introduce a somatosensory part in the BMI. So artificial somatosensory feedback should be delivered to the brain. And so in the lab, we don't work on humans, as I said, so we work on mice. And what I'll try to show you is that we can study this also in mice. And actually, you'll see at the end of the talk and in Zineb Ayatou's talk that we're working towards having a mouse feeding herself. with sorry, I'm losing my words. So this is not from the lab, but obviously it's from a crazy French movie called Human Nature. But basically in the lab we're trying to teach a mouse to feed herself while using, in fact, a fake mouse arm that she's using as a neuroprosthesis. Okay, so that's our general framework for our work. And so our question is really, how is somatosensory feedback used to adapt motor commands, both for natural skills but for BMI skills? And so first the question is, where in the brain could this be taking place? And so in the rest of the talk, I will focus on S1 and M1, so the primary somatosensory and primary motor cortex. And that's what people have been doing on humans. I mean, some people do, like Richard Anderson, explore other areas. But there is a consensus that for voluntary, very fine movement, dexterous movement, S1 and M1 at least are very heavily involved. And this is true in humans, but true in the mouse as well. And there is a lot of literature on this. Okay, so how does it look like on the mouse brain? So this is the primary somatosensory and primary motor cortices in the mouse. And so especially we take advantage of the whisker system in the mouse, which is a very important sensory motor system for this animal. So the whiskers are these long hairs on the snout of the animal, and they use their whiskers to explore their environment and to recognize objects. So there is a huge part of the cortex that is dedicated to these whiskers. So for us, it's a nice advantage. Okay, so the way we're doing our studies is by building a prosthesis. And so we use motor activity from the motor cortex in order to control a prosthesis, and I will come back to what I mean by that. And then we provide feedback to the animal about the state of the prosthesis by sending sensory feedback to S1. So it's very much like what is being done in human experiments. But with the mouse model, we hope to be able to test things in different ways and in more tractable ways. So our specific questions that I will talk about today is concern the somatosensory feedback part of this loop. And so I will concentrate on trying to show you how we should write sensory feedback information to the brain, and especially whether we have to consider specific spatial rules of providing this feedback, and also temporal rules. So this loop, so again, it's very similar to what is done in humans. For now, there is mostly one study that has been shown implementing this in a human from Charlene Fleischer in the laboratory of Robert Gaunt. And they implemented electrical stimulation in S1 coming from two sensors on a robotic arm, and it helped the human to manipulate objects. But this is still a very early prototype. Okay, so I will talk about three different studies, and the first one is a simple study. The question is really first to check whether the mice can perceive spatially distributed cortical stimuli that is provided to the cortex. And so this is going to be in open loop. We're really doing a perception, detection, discrimination experiment. So there is no motor control. And it's been done by Henri Lassagne when he was a PhD student in the lab. So we have this big chunk of the cortex that is representing the body of the mouse. And you see here that with a simple staining that it's even engraved in the anatomy of the mouse brain. So we can see the different parts of the body of the mouse represented in S1. Here you have the barrels corresponding to the whiskers of the animal. And so the question is, so where do we want to stimulate? Is it important where we stimulate in this map? So we decided to approach this by stimulating with optogenetics, because we have the tools in the mouse to do this. And so it allows us to activate excitatory neurons with light. We have channel rhodopsin in these neurons. And in what I will present for the rest of the talk, the task of the mouse will be to detect a photostimulation. And so there will be green areas that indicate a rewardable zone. And so when light, optogenetic light, is shown in the rewardable zone, this means that the mouse can lick and then it can get water. So the mice will lick for reward when the stimulation is in the rewardable zone. And so we attempted this with just one spot of light, so testing whether the mice could lick reliably for a spot of light in the right zone. And this is basically what we found. So the mice learned to lick for stimulation in the right zone and not for sham stimulation. I'll show you in a few minutes another pattern of stimulation that we tested, which was made of several spots. So here we have 5 spots of light, but I'll come back to that in a moment. Right now, for the next few slides, I will show you a different design that we used, which is a rotating bar. So this is the optogenetic bar of light, and it can rotate in this disk. And the task of the mouse is to lick when the bar is in the rewardable zone, a sector here, and it's to not lick when the bar is in the red zone. And so the mice have to learn this. So this is one example of a trial. So you see the bar of light rotating on the cortex. And so initially the mouse does not lick. And when it gets to the white zone, the mouse starts to lick. And only when it's in the rewardable zone, the mouse actually gets water. And then the trial gets interrupted by a no-lick zone. Yeah, a lick in the no-lick zone. So that's what the mouse learns. So you see here the 20 trials of the first session for one mouse, and you see that. So you see the licks as dots, and many trials were interrupted by a lick in the no-lick zone, and a few trials. For a few trials, the mouse managed not to lick in the no-lick zone. But did not necessarily lick for the rewardable zone, so did not get water. So the first session, the mouse didn't get a very good performance, but after about 5 sessions, this is the pattern that you get. The mouse did not interrupt trials anymore, almost not, and it managed to lick for reward in the right rewardable zone. And you can even see that it started licking before in general. And this is something that we saw repeatedly. So if we look now at the lick onsets, we look at lick onsets because mice tend to lick in long bursts. And the lick onset is really signifying the decision of the mouse to lick. So if we look at that, we see that they actually start to lick when the bar is in the white zone before entering the rewardable zone. So we quantified all this, and basically, these are curves that basically quantify whether the mouse is able to lick in the rewardable zone and to avoid licking in the no-lick zone for our population of 8 mice. And so our conclusions is that yes, they can learn this, and we also we had not anticipated this, but the mice actually lick just before rewards, so they anticipate rewards. And we think this is due to the dynamic aspect of our stimulation. Okay, so to go back to the question of somatotopy, which is really what we wanted to address in this study. So we found that the mice could learn this task in S1. And so we asked whether they could learn this task in a part of S1 that would not be as continuous in terms of topography. So because in the barrel cortex, the map is quite continuous. It represents the snout. But here, you have a body, what we call body S1. And here it's much more fragmented so that you have a representation of the trunk and of the legs. And a stimulus in this zone might be jumping from one part of the body to another. And so we tested our rotating bar in this body S1 representation. And what we found is that actually the mice could not learn the task for this area. And we also tested it in PPC, which in the mouse does not have a clear topography. And again, with this lack of topography, we found that the mice could not learn to discriminate where the bar was in this disk. So we think this is a good demonstration that spatiotemporal continuity of these stimuli is really important. If it's disrupted, learning is impaired. And we also tested this with shuffled stimulation, although I did not show it here. So our conclusion of this study is that mice, yes, they can perceive these stimuli, and they can interpret them even for a sweeping stimulus, and they even learn to anticipate stimulus dynamics. Okay, so when we finished more or less this study, still we were wondering what the mice were really perceiving. So are they perceiving tactile stimuli or not? And this is a question that goes back to what Richard Anderson showed, which is that said that that humans, when you stimulate them in S1, they perceive tactile sensation. But for the mice, we cannot just ask the mouse to report. So what we did is we did a transfer experiment. So we trained mice in this task that I just showed you, and then we transferred the mice to a setup with a rotating bar, a physical rotating bar. And what Alexandre did is that he showed this bar, and he asked again the mice to lick when the bar is in the rewardable zone, and he chose the rewardable zone that corresponded to where the whiskers deflected correspond to the barrels of the optogenetic stimulation. So it's a matching transfer. And indeed, the mice could perform this task very well as soon as they were put in the setup. So they had a similar performance to the optogenetic task. If you invert the pattern and you now the mouse has to lick at a very different moment, a very different place in the barrel field, then it does not work. The mouse do not have a good performance. So we think this shows that there is really correspondence between the optogenetic and tactile stimuli. Okay, so I'll go to the second experiment I want to talk about. And so here, now we're going to look at the brain-machine interface. So we're going to introduce the motor command part and look at the closed-loop version with spatially distributed feedback implemented in S1. And this has been done with Amir Abbasi and Henri Lassagne. So this is the full closed loop. So it's very similar to what was discussed earlier with humans. So M1, we are recording activity from M1 as single neurons. And the firing rate of the neurons is translated to the position of a virtual cursor, which can take 8 positions. And so there's a correspondence between firing rate and position. And here it's very simple. There's no machine learning or anything. And the state of the cursor is provided to the mouse as an optogenetic pattern. And so there are 8 different patterns that can be shown. And here, so we did not use a sweeping bar. We chose to mimic the pattern of discrete deflections of whiskers, as a bar would be moving from the back of the snout to the front. And yeah, this is the pattern on the cortex. For the electrophysiology, we use very standard tetrode electrophysiology. We sort spikes online. Yeah. And so we use 3 neurons to drive the prosthesis. We call them master neurons. And we sum their activity. We just simply do a sum. And so we transform this into a cursor position. And so these are the patterns of optogenetic spots that are projected on the cortex corresponding to the positions. And so the mouse is licking. And if the mouse licks when the cursor is in the rewardable position, then it can get a reward. So that's the task. So in session 1, so this is an example mouse. In session 1, the mouse licks, but it doesn't get any reward because the timing is not right. In session 5, the pattern of activity has changed a little bit, and the mouse now manages to lick at the right times in order to get rewards. So the performance of the mouse increases over 5 sessions. And you can see that if we train the mice without feedback, they don't manage to get this task. So the cortical feedback helps to train the mice to in this BMI. So the question is, so what is important in the feedback? So in particular, is the spatiotemporal pattern of the feedback, is it important? So to test this, we shuffled the feedback. Sorry. Let's see if I can play this. So this is the original feedback I showed you, and we did a shuffled version where the spots are more random. So we lose the spatiotemporal continuity both inside each frame, so the spots are not aligned, and across frames too. And so with this version, the mice actually could not learn the task, which shows that spatial organization indeed seems to be very important for this task. Now we also wanted to look at what the mice are actually learning. So are they learning only to detect when the pattern is in the right place, or are they learning to move the prosthesis to bring it really to the right place? And so we looked at motor control, and what we found is that the mice, we looked at trajectories. So here you have virtual cursor position, and we found that indeed there was a ramping up in the cursor position well before reward, and which was found at the end of training and was not present if there was no feedback. So it seems that the feedback is helping to really have trajectories going to the rewardable zone. And this is just a quantification in different time windows around the reward. And yeah, so this is in terms of trajectory of virtual cursor position, but this position is directly linked to the master neuron activity. So we asked what is happening with the neurons, because we're summing the activity of 3 neurons. So are these 3 neurons all participating or not? And so we looked at that, and we were a bit surprised to find that initially the neurons might have similar firing rates, but after training in session 5, there was 1 neuron that really took over. And this is for one example, but it was true also in the population. There's really 1 neuron that seems to be dominant, so the dominant master neuron. And this we we had not really expected, but it's in line with other results in the literature. And so they learned to move the cursor, but they also learned to synchronize licking with the opportunities for reward. And this we showed by looking at the correlation between onset of licking and entry into the rewardable zone. And again, we found that this was developing with the bar feedback condition, so that after training they synchronize licking at the right times, whereas it's not developing if there is no feedback. Okay, so the conclusion for this study was that mice could learn to control a virtual prosthesis by modulating M1 neuronal activity, and that when we provide optogenetic feedback, the spatiotemporal continuity of the feedback organization was really critical. Yeah, and we actually think this is one of the first demonstrations of a functional role for S1 somatotopy. So it's not only something that is inherited from development, but it's actually used in this case for extracting relevant features. And maybe it's used especially in the context of sensory motor learning, more than perception of sensory inputs. Okay. So I will go to the third part, the third study, which is an ongoing study. So as you'll see, we don't have full results yet, but I just want to give you an idea of what we are doing. So, and now the question is that we wanted to investigate the temporal aspects of somatosensory feedback. So when we do natural movements, so if we want to grab something, so let's say I want to grab this microphone here, I'm using sensory feedback as I do that, and somehow in my brain there is, my brain knows at what latency this feedback is coming. It's in the circuitry. And so if the feedback is coming too early or too late, I might be impaired in my movements. So if I saw my hand 2 seconds after it actually has moved, then I would not be able to grab something. I would probably overshoot, etc. And so when you want to provide feedback directly to the brain, you have to decide at which latency you're going to do it. So this is the question. And so in this system, in the whisker system of the mouse, so we estimated that the physiological latency between spikes in M1 that drive movement and consequences in terms of sensory feedback is about 50 milliseconds. So that's kind of our reference. And so, so for this experiment, we decided to go back to the rotating bar because it was too difficult to design the task with jumping spots, in fact. And so we went back to this rotating bar. But contrary to the first part of the talk, now this bar is directly driven by neuronal activity. So we're not in open loop, we're in closed loop now. And here, so the way it's set up is that we have one neuron that makes the bar move in one direction by a few degrees, and one neuron that makes the bar move in the other direction by a few degrees. And so the mouse can rotate the bar by modulating activity of these two neurons. And other than that, the task is exactly the same. The mouse has to lick when the bar gets in the rewardable zone. And so what happens is that we can modify the latency of the optogenetic feedback compared to the cursor position, the rotating cursor position. And because we, Henri Lassagne designed a very efficient closed loop system, our minimal latency is 5 milliseconds, which can be updated every 1 millisecond. And so we can choose between, so we're trying 3 latencies right now, which are 5 milliseconds, so faster than physiological feedback, 50 milliseconds or 500 milliseconds. But I'll mostly show you for now the 50 millisecond and just a little bit about what happens when we change. Okay, so these are the same graphs as before. The only difference is that the mouse is now driving the prosthesis by M1 activity. So you still have, in session 1, you can still, you can recognize that there are many trials that are interrupted by licks in the no-lick zone and no rewards. In session 12, the mouse has learned, so there are many trials, so very few trials interrupted, and many trials in which the animal manages to bring the bar to the rewardable zone and can lick in the rewardable zone, get water, and perform the task. So the performance, in terms of number of rewarded trials, the performance goes up over 12 sessions with this training at 50 milliseconds. So we also looked at different parameters as we had done in the open loop experiment. And in particular, we looked at the lick onsets, and we see that again, the mouse starts to lick before it enters the rewardable zone. So we still have the same anticipation of the coming cortical stimulation. So the conclusions for this licking behavior are exactly the same as in the open loop. Now, that's just a quantification for the population of the lick onsets in the different zones. So what about the motor control now? So the motor control, we look, to quantify this, we look at the trajectories over time. So each trial can last up to 20 seconds. And so you see here the different trajectories for session 1. And you see that it kind of looks like a Brownian motion at first. So the bar is moving not in a very coherent way, well, not consistently across trials, so it's just exploring. And there are very few excursions to the rewardable zone, and so very few rewards. In session 12, we can see that there are more stereotyped movements. And so here you see that many trajectories, many trials manage to reach the rewardable zone and to stabilize either in the rewardable zone or very close to its border. And there is a lot of licking, so lots of rewards there. So the mice learn to bring the bar to the right zone and to lick there. This is a second example where you see a very wide array of different trajectories in session 1. And here in session 12, the mouse chooses to send the bar to the rewardable zone by going to one direction, much more stereotypically, and stabilizes next to the rewardable zone here. And we see this in many of the mice. They actually choose to concentrate on one side. They don't explore necessarily the two sides. They seem to develop a stereotyped trajectory. Okay, so this slide is a bit complex, but I want to show you a little bit the activity of the neurons below. And so here we have the activity of neuron A, which pushes the bar in the rewardable zone, and neuron B in the opposite direction. So in magenta, you see that neuron A fires before the entry, which is by construction of our algorithm. And neuron B has a lower activity here. And what's interesting is that in session 12, what we noticed is that neuron B seemed to develop a high activity just after entry. And this has the effect of slowing the bar once it enters the rewardable zone. So it's like applying the brake, if you want. And we saw this quite consistently. And So this is quantified on this example by looking at the activity after entry relative to before entry. So you see that across the 12 sessions, there is an increase of this modulation. And when we quantified it in the 6 animals that we tested, we see that indeed between the first and the last sessions, there was a strong increase in this slowing down mechanism. This seems to be the most important change that we have, that the opposing master neuron is modulated. And so this reminded us of our dominant master neurons. It's like there's one neuron taking over the task. And and that allows to get more time in the rewardable zone. So, okay, so this was the main results I wanted to show with this 50 milliseconds latency. I'll just show you very briefly what happens when we try 5 millisecond latency. And so here we trained one mouse with a 5 millisecond latency, so the feedback is coming before what the brain should be expecting, if you want. And so we see here in session 1 quite variable trajectories as expected. And in session 12, the mouse has developed a different set of trajectories. And so here it chooses mostly to bring the cursor, the rotating bar, to the rewardable zone on one side, and to get a lot of rewards by exploring on this side. And so the mice can do it, but for now, we found that learning was much slower than in the 50 milliseconds case. And this has to be confirmed. And in terms of activity of neurons, it's a bit similar. We found that the modulation of activity that we had found for the 50 milliseconds case is actually much less for the 5 millisecond latency. So it seems that changing this latency is altering the behavior of the mice. And yeah, just my last data slide is this one. So here we have tried again the transfer experiments. So we wondered, now that the mice have learned to manipulate this optogenetic feedback, what happens if we show a real bar? And so it's the same experiment as before, but done on an animal that went through the closed loop training. And so the animal is moving the bar and manages to stabilize the bar in the rewardable zone as soon as the first session where we try it. So we were quite impressed by the performance in this first session. We hadn't expected the experiment to work so well. There is a poster from Alexandre Tolbaum on this, so feel free to go talk to him. He will show you some more results. So the conclusion of this third study was that mice could learn to control such a sweeping bar by modulating single spikes of 2 neurons. And we are currently completing our data set to have results on the 500 millisecond latency, which interests us a lot. Oh, sorry. What did I do? I pressed the wrong button. Okay. Before I stop, I want to just give you 2 snapshots of current directions in the lab. So one is a study that is performed by Anton Dogadov. And the idea behind this project is to use mesoscale cortical activity to control a BMI. So it's very much in the same line as trying to use functional ultrasound imaging or other signals that would not require electrodes, but that are at the mesoscale. And so what Anton did is that he imaged the activity of the sensory motor cortex with calcium imaging, and he trained mice to generate a wave of activity in the sensory motor cortex in order to drive a BMI. So he has a poster about this. And finally, So Zina Bayatu will talk about this project, but just to give you a brief snapshot, Luc Estebanez in the lab is also developing ProMice, which is this robotic forelimb for a mouse, equipped with different motors and touch sensors. So now we have a physical prosthesis, whereas everything I've showed you before was with a virtual cursor. And what she will show you in a moment is that she can test embodiment of such an artificial forelimb, and we think it will be important to have a mouse model of embodiment. And so now you'll see that they're very close to training mice to feed themselves with this robotic forelimb. Okay, so to conclude, I showed you that we developed an ultra-fast closed-loop BMI in the mouse with a robotic mouse forelimb neuroprosthesis. And I hope I convinced you that we can also decipher mechanisms of sensory motor skills using this BMI. So topographical constraints are important, timing could also be important, and we hope this is going to be useful for the human community as well. And so here are the people involved in the project, so the whole team. This was actually taken at the first joint conference NeuroPSI TCCI last year. And so I showed the PhD students already implicated in the project, but of course Luc Estebanez and Daniel Schulz and Isabelle Ferrezu contributed as much. Thank you very much.